Hi, my name is Chantel Carroll, and the title of my presentation is called Antibiotic Resistance Discovered in Toxic Environment. So before I get to the experiment I conducted, I provided a little bit of the background. So acid mine drainage is a metal-rich water formed from chemical reactions between water and rocks containing sulfur-bearing minerals. It forms sulfuric acid, which then dissolves heavy metals such as copper, lead, and mercury. The problem with acid mine drainage today is it causes contaminated drinking water. It disrupts growth and reproduction of aquatic plants and animals, and it's one of the most serious water pollution problems today. The picture I provided is actually a picture of an acid mine drainage that was taken in Ohio in 2014. Antibiotic resistance occurs when bacteria and fungi develop the ability to resist a drug that was created to harm them. It occurs naturally, so it's not something that we can control. Antibiotic resistance can be tested in the laboratory by using agar plates and antibiotic discs. A zone of inhibition is the circular area around the antibiotic disc where the bacterium does not grow. So if there is not a zone of inhibition, that means that that bacterium is resistant to antibiotics. The problem with antibiotic resistance is it causes infections become harder to treat. It leads to higher medical costs and increased mortality. So the experiment I conducted was unique bacterium was isolated from the acid mine drainage located in Bice Park, which is in Allegheny County. The DNA was sent out for shotgun sequencing. It was assembled into a genome on kbase.us. Antibiotic resistance was tested, and it was hypothesized that the unknown genome, the unknown bacterium, is resistant to penicillin and ampicillin. So I provided a diagram of half of the methods that was used in this experiment. So acid mine drainage was plated on R2A plates. A single colony was isolated, the DNA was extracted, and then sequencing was done. And genome annotation is where K-base comes into place, and then a hypothesis formed, and the hypothesis was tested. So R2A agar was obtained. 2 plates were labeled pen for penicillin. Multiple penicillin discs were placed into the agar with a concentration 0, 1, 5, and 10 micrograms. The disc concentrations were also labeled on the plate. 0 micrograms was labeled B for blank. The same happened for ampicillin. 2 plates were labeled AMP, which stands for ampicillin. Multiple ampicillin plates disc were placed into the agar with that concentration 0, 5, 10, and 20 micrograms. The disc concentrations were also labeled on the plate, and 0 micrograms was labeled B for blank. The control plate was labeled plus control, and there wasn't any antibiotic disc added to this plate. The genome was added to all plates, including the control, incubated for 48 hours at 30 degrees Celsius, taken out of incubator, and determined if there were zone of inhibition on the plates. kbase.us gave me genes that would be present that are present in my genome. I found out that Beta-lactam, Vancomycin, and Macrolide were present in my genome. So the Genome Tree was provided to me by kbase.us. It actually showed me the closest related relative to the genome I had, to the bacterium I had. So CC genome RAS is actually the genome I've had, and its closest related relative was Brevibacterium linens. kbase.us also provided me with an fast ANI software, which stands for Average Nucleotide Identity. It was completed with my genomes. It was completed with my genomes closely related relative to determine the genomic similarity between the two. ANI's purpose is to compare DNA to determine relatedness. So I was able to conclude that bacterium was not inhibited by penicillin. So my antibiotic, or my bacterium, is resistant to penicillin at the concentrations 1, 5, and 10. Bacterium is inhibited by ampicillin. So there are zones of inhibition at 10 and 20, which shows that the bacterium is sensitive to ampicillin at 10 and 20, but not at 5. So at some degree, it is resistant to ampicillin. So the zone of inhibition was measured at 10 and 20 micrograms. So at 10 micrograms, the zone of inhibition is 0.4 millimeters, and at 20 micrograms, zone of inhibition is 0.6 millimeters. So I was able to conclude that the genome contained at least one of the beta-lactam resistant genes, so one was able to test for ampicillin and penicillin resistance. R2A agar was used because it's used for detection of heterotrophic bacteria in water. Blank disc is the control. It is to show that the disc themselves do not inhibit growth. Disc diffusion method is used because one can tell how sensitive the bacterium is to the antibiotic by the zone of inhibition. The larger the zone of inhibition, the more sensitive the bacterium is to the antibiotic, which we could see because at 20 micrograms on the ampicillin plate, it was bigger than the zone of inhibition on the 10 micrograms. Using the disc diffusion method, one can use one agar with up to 4 different antibiotic discs inserted into it, so it becomes a cheaper method to use. Concentrations for ampicillin and penicillin were different because the minimum inhibitory concentration was trying to be determined. The unknown genome's closely related relative is also called the lactic acid bacteria. It helps ripen cheese such as Limburger. It is also a bacteria that causes feet odor. A bacterium that creates foot odor produces isovaleric acid, which can survive in acid mine drainage. The fast ANI software, I showed that it did not identify the organism and that it may be a novel species. I say that because ANI software showed that the two bacteria are over 91%, but it did not meet the accepted threshold of 95%. So they are closely related, but CC genome may be a novel species. The unknown genome was not identified in this experiment, but it has shown that antibiotic resistance has occurred with penicillin. The next experiment done with this genome, it may adapt and become completely resistant to ampicillin because the zone of inhibition was not that big. Future experiments, you can test Vancomycin resistance by putting the genome on a brain heart infusion agar, abbreviated as BHIA plate containing 6 micrograms milliliters of Vancomycin, incubate and observe for resistance activity. Or you can try higher levels of penicillin concentrations to try and determine the minimum inhibitory concentration. Thank you for listening.